guys, like we are all the students of Tlageshwar as well. So, uh, as he said, that I've been doing a little bit different during my postdoctoral studies as a mechanosensation studies on bacteria, Pseudomonas aeruginosa. And as I was moving back to India, Thermo Fisher was one of the company who was actually offering a position which is related to structural biology. So I was happily um, eager to join this. And once I've joined, I've learned a lot of things as well as about what's happening in the current trends in structural biology, especially in terms of cryo-electron microscopy. So I think most of you would have already got this introductions on cryo-electron microscopy through the entire day. I've seen like wonderful speakers. Um,
Okay, just Kumar, just a second. Actually, are you comfortable if you start the recording? Actually, record your session. Are you
yeah,
okay with that?
absolutely, absolutely.
Okay, okay, fine. Yeah, thank you.
Yeah, as you might be aware, I mean, you would have learned a lot from professors, previous people like Natesh and others who have been constantly involving in the application side where they use the cryo-EM. While I am from the company Thermo Fisher, where they play a decent role in developing the tool in such a way that it can be used that can be used for many of these biologists, especially in structural biologists. I hope I will be trying to convey some of the historical perspective of like how these tools are developed. Since most of you guys are from engineering background, like you guys have some sites, it would might be interesting to you, and some of you guys are from the. I mean, most all of you are actually bound to learn this life science and structural biologies at some point. You would be also a little bit intrigued by what kind of developments that we have gone through. Um, so with further thing, I will just get into the topic. So this is one of the constant slides that we keep showing up from Thermo Fisher, and we are kind of just have to show must show a presentation slide. It's like Thermo Fisher being a world leader in supporting the research in all kind of fields. It has a big conglomerate of companies which have been able to provide decent support for many of the researchers. So the most iconic tool that Thermo Fisher has been happy to give delivery to the researchers is the electron microscope, and we are proud as a company to actually able to participate in most of the engineering developments and tool development, software development for the Cryo-EM, and it shares almost like 60% of its revenue for R&D development of electron microscope itself. So what is this electron microscope is basically, it's just for some of you to understand this, like uh, it's like if you are taking a if you want to see a rock on a moon with your naked eye, the same kind of magnification you're going to see through the microscope, just using the electron microscope, electron beam, you're going to see the same kind of magnification at that level. So pretty much you're going to see electronic clouds, or the atoms in a near atomic level. So uh, so even the slightest disturbance in any form is going to be causing a major issue in focusing, and that's where the engineering part of the Thermo Fisher team has played a huge role in developing a very stable tool for life science people who are life science. So this electron microscope had a humble beginning around 1949, where it was like somewhere it was built on a desktop. Now the actual the high-end electron microscope, the height is somewhere around um, about 3 meters tall, so it's quite huge in stool. And in 1997, the FEI got the Philips actually started out building the electron microscope, and FEI took it over in 1997, and in 2016, Thermo Fisher acquired it. It's around the same time where resolution revolution happened. I will explain to you a bit later about what has happened in 2017. So just to give a simple um, diagram image of this is the old generation electron microscope, where you can see on the right side. So basically, it's just like instead of the light source on the top, what we have is an electron beam, which is passing through the condenser lens and the objective lens, and once it passes through the specimen, you get a fluorescent image, and it's as simple as the light microscope itself. But the only complication is electron source. We are going to use electron source. And why do we use electron source? Is because it's cheap to generate an electron source, and its most important thing is it has a charge nature, because of which you are able to bend the electrons and at your will, um, basically through extreme coils of coppers and different circuits. You might be able to bend this electron so you can precisely and perfectly focus it on the specimen. So this is the simplest version of the electron microscope. As you can see, there's a source which produces the electron beam, and then there's a lens, and then there is going to be a detector. The source is going to be this is a this is a primary tool that we basic tool that I'm showing here. A thermoionic filament is there, where you just heat the tungsten filament, and then it's going to release electrons, which is going to be um, amplified, like condensed into a very small range, and then uh, this is going to give you a lot of electrons, which is going to be manipulated in a way that it can be precisely hit the samples. And there is a lab 6, and then the recent one is XFEG field emission gun, which is a very thin, which is a very pointed tungsten, where you can use it to generate a field where a negative field is sorry, a positive field is applied, in which the sharp edge is just inserted, so just the electrons are pulled out of it without getting heated, so you have a very coherent electron source. So this is how a typical field emission gun. So there's going to be a coil of coppers in through the hole. Just right next to the hole, you're going to insert the sharp edge of the tungsten. So, and you're going to give a very mild current on it, and once the 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 edge is going to be insert the hole over here. Just a sec. I'll use a pointer. So on the point when the sharp edge is going to be here, the electron source are pulled, and it's going to be creating a very coherent electron beam. So this is going to be a lot of information. You don't have to know every bit of it, but all that you have to know is there are multiple 3 condensers currently being used at a high-end electron microscope, which gives you it's a kind of a switch or a knob which helps you tweak the electrons beams in a way that you're going to coherently or make sure it is as um, same as possible, and as uniform as possible, and as focused as possible before hitting the samples. And later on, which after the condensers, there will be the samples are focused, the electron beams hit the sample, and then you kind of again condense the diffracted electrons, and then you again uh, put it focus it towards the uh, focus it towards the detector at the bottom. So nowadays, most of is all of it literally. After like maybe 10% of this is manually done, rest of which is all done by computerized. All this condensers are adjusted with extremely highly uh, by powerful computers, and even the samples which is loaded over this socket, or like the hole over here, is also computer it's also practically operated by the computers. So this is a very basic model of cryo holder, where the copper grid is placed in a hole, and then you just insert it through this hole, and right at the direction, or the direction of electron beam, you place the sample. So basically, you can actually um, tilt uh, tilt the cryo holder, the holder, in XYZ direction. But most of the researchers would actually prefer just to move it in XY direction. Just you're going to do this. In some cases, you can also tilt in alpha, beta, and rotations like that, which will be helpful in collecting different data from different angles of the same samples. So finally, when it comes to the detector, there's a multiple detectors have been developed, and the recent one is the Falcon. So the Sita, Eagle, Velata, Flucam, Sita, and Velata, Aureus are all like CMOS camera is basically um, what you have is a sandwich of diodes, on top of which you place a fluorescent cam. So when the electron beams hit the fluorescent cam, the electrical changes are observed by this camera, and these electrical impulses are Fourier transformed to get a image. But the Falcon 4i, which is a direct electron detector, which has a beautiful engineering in it, um, but so what happens is, every single electron hit the sample, it's going to be observed in this direct electron detector, and every single electron is actually counted at the exact position where it is counted, and each and directly it is going to create an electrical impulses, and which is going to be Fourier transformed into an image in the later part. So what happens is, if you actually have this Falcon camera and take a photo, I mean, in some future, um, if you have this camera on your phone, when you're going to take somebody's picture, and you can zoom in all the way through the photo, and you can actually see the cells and you know atoms on somebody's face. It's going to be that big deal. But right now, the size of this Falcon camera is somewhere uh, to be like uh, almost like a box of one 1 feet by 1 feet, of that big of the camera. Hopefully someday in future we might, once we have the quantum mechanics development in the quantum mechanics, maybe we will be able to fit that in our mobile phone as well. So why did we go through all this? Just to initially, this all these things were used, actually, why did we go through this is because as a human, we were always interested in understanding how the life works at some point, and we wanted to know. So we did microbiology, we did molecular biology, and still there was a lot of questions remained, and many of the biophysicists, or like by life science community, has tried to understand the key, or the major information about the life actually involves in the structure of protein or the biological molecules. Although this electron microscope has been mostly developed and utilized by material science, biology was not far behind to adapt it, and in recent years, now the cryo-electron microscope has become the major tool to observe the structural change, which which indirectly determines how a protein or a biomolecule functions, and how these function of all these biomolecules synchronize together to maintain the cellular organizations and even cellular function. And in some sense, it dictates what we do, what any life would do. So the previous methods that was used was quite difficult as a crystallographer. Although I was little bit hesitant at initially to accept the to power of this cryo-electron microscope, I have to agree to this, that cryo-electron microscope has indeed reduced the time that we spend on crystallization and optimizing protein purification and a lot of other things. So we could get structural information in a very brief time. So this is a general idea of what. Is there anyone asking questions? I'm unable to see.
No, Kumar. You can proceed. Actually, I'm just
Oh,
happy.
okay. So Cryo-EM, so these are the major tools currently available for understanding um, a protein, a biomolecular structure. So X-ray crystallography, which has been widely used before 2020, 2019 maybe, which was, you can get a resolution of 1 angstrom, 1.5 angstrom resolution of protein molecules. But the limitation is have you cannot process a protein or like get a structure of any biomolecule. The best you would get somewhere around 200 kilodalton. The majority, the median would be somewhere in third, I mean below 60 angstrom, below 60 kilodalton, and the resolution is pretty good, but you cannot go to the biomolecules of larger size. And NMR, which is also a prominent structural biology technique, has its own limitation, where you can see the biomolecule size of 30 kilodalton and slightly above that, but majorly it focus on small molecules. And the resolution is not that great, but you will get a dynamics of the uh, biomolecules. But cryo-EM somehow it's actually it's just showing 60 kilodalton. Actually, it's gone down to 40 kilodalton, and the the size of the protein is almost like 10 megadalton. You can solve the protein structures or the biomolecule structures, and the resolution has come down somewhere 1.2 angstrom resolution. It's pretty close to the X-ray crystallography resolution. That's the recent developments happening. So what do we have is Cryo-EM tool that's at 300 kV, um, and this is the Cryos tool that's best in the um, best tool that EM tool in the world. I might be little. I'm not. So okay. So this is used for the biological studies. What are the questions asked for? You can pretty much ask basic questions on biochemistry and how the biological functions happening. And the tool has been designed in such a way that the microscope, people do not need to be an expert in the microscopy. All that the programs are AI guided, and it can easily guide the people user to study the biomolecules and majorly focus on the data that they collect from the tool. And this is a cost effective in some sense, although the numbers are going to be in few crores. If you take any other structure solving tool, it's going to be in few hundred millions if you're going to solve just the structure of human proteins. And this is actually like in couple of millions, and then you will be able to solve the structure, and it's going to be pretty cheap compared to what you are going to invest on the research itself. And the output is quite dramatic, as you will see as I will be explaining the role of cryo-electron microscope during COVID. So the resolution revolution happened somewhere around 2017. The upper 14 was the structure which was solved at 1.62 resolution. So this actually I need to update. This has reached to 1.15 angstrom resolution with the current cryos. That's cryos G4 and Falcon 4i is the camera that we are currently using. So during the resolution revolution around 2017 onwards, there has been major breakthrough, and the first time researchers have using the material science tool, where they introduced a cryo concept in it, so that they can cool the sample, so they can expose the sample for a longer time, which will help them in solving the structure of protein. at a high resolution. So around the same time, we would actually, around the same time, the cryos, which is like 2007, the cryos, you can see that Thermo Fisher, the FEI has been investing a lot in the development of this tool. So, moment the DED electron, this direct electron detector, was launched, as you can see, the resolution fell somewhere around 4 angstrom resolution, and then around 2017, we created 300 tool with selectries, which I will be explaining in later part. Selectries is uh, you're going to select a band of electron beam, so you can get a better image. So moment we reached there, we got somewhere around 1.15 angstrom resolution, and the from that time onwards, we have been consistently getting atomic resolution biomolecular structures. The three major people who have been involved in the development of cryo-electron microscope, these three people, that's Joachim Dobschert and Richard Henderson and Joachim Frank. So all three of them took part in the three major regions of the development of cryo-electron microscope. So Joachim actually was involved in vitrifying the biological sample, so credit goes to him. And then Richard Henderson, who was involved in the data acquisition, and Frank was actually in. He may give a major contribution on 3D structured reconstruction. So all three of them got Nobel Prize for 2017, which is like 10 years from the revolution actually happened. So several people after them, they came out and showcased the power of Cryo-EM. Um, so this is the development or the historical perspective of we in 2007 Titan cryos was used. That's 300 kV field emission gun, FEG stands for. So they had an autoloader, which means you can load the samples by a motorized mechanism. And then in 2017, we also had a 200 kV, which had pretty much similar parts like Titan Cryos. Um, but the resolution wise it has been a little bit compromised, but the usage wise it has become more user friendly. And then once we developed the user friendliness in glaciers, we could also develop Cryos G4, which was 10 times faster than the previous Titan Cryos, and the size was quite smaller. And currently Thermo Fisher is having Tundra and a Glaciers 2 version, which has been just launched a couple of months back. So Tundra and Glaciers are Glaciers, and Cryos are the three major tools that we offer to life science community currently, so that they can do structural based studies on their for their research. Um, this is just a video of how the electron microscope works. So the biomolecules is quickly frozen at minus 180 degree by plunging into ethane, and after which we put it in liquid nitrogen. So then the beam of electron passes through the sample of the protein. So you can just pick up small particles. You can see it on the from the detector. And as you can see in a Iron Man movie, if you have seen in the second part, there's a he will try to invent some kind of uh, new element. So in that, a 3D hologram of sorts will be created, right? So similarly, we create a 3D hologram of each by taking several snapshot of the uh, same sample with multiple particles of the same sample. So you could do a 3D reconstruction, and then you get a protein structure of a protein. It's very simple as it is said, but the kind of as you can understand, like every part of it, um, to develop this tool. Um, i- so we have actually calculated the number of parts that has gone through. That's electronics. It's almost like 3 Tesla cars. If you the same number of electronics that goes into 3 Tesla cars has been incorporated in one cryo stool, so that you get this kind of an images. So, is it available in India? Yes. Dr. Vinod from NCBS was one of the pi- pioneer in developing in who worked with the Richard Henderson's lab. He's working currently at NCBS, and he was the first one to install cryo stool in India. So he has been quite often using this tool. Um, so what he has is like sort of what we have to do with him is like we provide most of the tools. Like he has been constantly buying all the tools that's required, like Talos L120C, like a slightly smaller version of cryos. So the electron volt is 120, so it can be used to optimize the sample before going to the cryos. And then we provide basically all the tools for getting the best resolution, starting from protein purification all the way to the data interpretation. Each step of the way, we have some contribution or other. So this is the general workflow. So you have the protein purified or biological sample purified. In this case, from now on, I'll just stick with protein. So if you have the purified protein, and then you run the gel, SDS gel, and then you screen the or just load it in the Talos L130C, which is a smaller version of electron microscope, and then then you can actually monitor it, and then you vitrify it again. You can go back and monitor how the particle distributions and how your samples are behaving. And later on, you can go through the glacios, which is the higher end version, and then finally you can go into 300 kV to solve the structure. And nowadays 200 kV is as good as 300 kV earlier version of like G3 version of 300. Still you can get a high resolution 3D constructed proteins. So in some cases, you might want to see the entire cells in its native state. So you might have to process the tissue a bit using a fib milling system, and then you can directly place it on um, the cryos. So I will explain a little bit in some of this in detail as I go. So currently, what do we have is the best resolution is 1.15 angstrom above 14. It's as good as it can get with cryo-electron microscope, but still there's lots of research going on um, uh, with all over the world, especially uh, yeah, on life science side, where they're trying to improve this quality, or can we push it beyond 1 angstrom resolution? That's the challenge that we are currently focusing on. And of course, the larger the protein is, like 10 megadalton poliovirus structure was able to solve. And then the smallest protein is like 40 kilodalton, which is quite challenging, um, even in some cases of crystallography. Still we are able to get cryo-EM structures with that. These are the some examples of practical examples of what the cryo-EM has offered. So 200 kilodalton GABA to 1.7 angstrom resolutions, and human hemoglobin 2 angstrom resolution, which is 62 kilodalton. And basically, um, you are getting in the range of atomic resolution, which is like 2 angstrom resolution. You are able to get the structures of protein. So this is just a comparison of how 100 kV, 200 kV, and 300 kV tool works. So the 100 kV would, if the best resolution you get is somewhere around 3 point angstrom resolution, 3 angstrom resolution, 2 point angstrom resolution for GABA, proteasome, and above 13. So once you have glaciers too, without selectors, you already, that's 200 kV. You already get like 2.8 angstrom resolution, which is like almost like 6.6 angstrom resolution you have shifted. And then moment you add selectors, you are already in the uh, 0.2 angstrom resolution, and as you can see 300 kV you are around 1.7 angstrom resolution. And for upper 14 you can see that you can shift it from 100 kV. If you see at 300 kV with the selectors, you will get around 1.2 angstrom resolution. There's sometimes a dream in case of many of these crystallographers when they work with this kind of protein, like GABA or proteasome, which are quite huge molecules. And at some point it was almost considered impossible. Some people, very few, very few success rate has been there. But now with Cryo-EM, 1.2 angstrom resolution for GABA is quite common. So with Cryo-EM, you can study the native function and the dynamics of the proteins, and you can find low affinity binder molecules, which plays a crucial role in determining how the biomolecules works. Um, so in other techniques, you need a high affinity binders, but in Cryo-EM, you can even process low affinity, and post-translation modifications are quite crucial, and it can be easily observed with Cryo-EM. And it needs a minimum processing compared to other techniques. And the amount of samples that you need to create, develop, or like produce for the experiments are in few microliters. And uh, there is no crystallization and reduced construct and optimization, and pretty much everything. Once you have the sample, rest of it are more like automated things. You can collect the sample automatically, collect the data automatically, and then even reconstruction of the data also is quite automatic. So Cryo-EM has been quite useful during the COVID. As you can see, in November of 2019, which was where the first case was observed. From that time on, just within Jan 17th, we got the first structure of China announces there's a coronavirus outbreak in Jan 17th, and then the first structure was solved at Feb 19th. It's like just over a month we were able to get a protein structure, which was almost impossible with any other technique. And then later on, we were able to get SAS in a bound structure. Uh, to the receptors, and then in the phase 1 of clinical trial we were able to see, which helped them to develop the phase 1 clinical trial by March 2020, which is like record breaking speed at which Cryo-EM has helped in understanding the structure of AS2 and the receptor binding proteins. And over the um, yes, is somebody's having a question?
No, Kumar, there was a mistake, I think.
Okay, no worries. Um, yeah, please feel free to interrupt me anytime, and just make a noise. I will be happy to answer any part of the question. Otherwise, I kind of keep going through the slides after slides. Um, I find it very interesting as I keep explaining, and I keep forgetting like others are also listening. Sorry about that. So feel free to interrupt me at any time. Um, so around May, we got some more structural informations, and all this during the entire year, like within 6 months or 7 8 months, you are actually able to get most of the information about the coronavirus and how they interact with um, humans. And we were able to see how the vaccine candidates are actually binding to the spike proteins and stuff like that. So uh, we were we were quite happy from us. Thermo Fisher, we were quite happy. We were able to contribute significantly during this development, not just developing any kits, but also directly involving in developing vaccines. Some of our buyers, or like people who vaccine developers like Moderna, Pfizer, Novartis, Astrogen, Serum Institute of India, and most of them are actually like they are all a big they keep using our products, Cryo-EM tool, um, and we are quite happy about it with the way we are trying to support the scientific community. So the recent developments are majorly focused on EPU. Um, eh, so what EPU is just a software that we have inbuilt to the computer, so it can interact pretty in a very well manner with the Cryo-EM tool. So which actually helped us in solving a structure of tau protein. Um, in a matter of 6 months, we were able to solve 76 structures. So this was quite Loustam et al. Actually, they solved the structure, 72 structure in just less than 6 months, and they were able to publish Nature paper. And if you compare to our competitors or any other uh, electron microscope developers, that they wouldn't even so overall entire lifespan they would the number of structure published are less than 70 actually, but we were able to get in a 6 month from one lab from we were able to get 76 structure and they were able to publish Nature. So that's the kind of output that someone could expect from um, by just making a development in software. uh, so this is another uh, developments of how GPCR has been GPSCR studies have been heavily dependent on Cryo-EM currently. So several structures on an average like 4 angstrom, 3 angstrom um, structures have been on a regular basis people are solving. This is from Patrick Sexton's lab from Monash University. He has been on a regular basis. He has been getting a structures, 3 angstrom, 3.5 angstrom resolution of GPCR structures. So this helps in understanding many of key things about protein structures itself. So this is one interesting thing that I wanted to highlight is like recent time Cryo-EM has actually overtaking sequencing itself. So this is the lab like Calber lab. They have been working on virus particles. So one day they are looking at worms, which has turning black and dying. So they, being a virus lab, they just wanted to isolate the virus. So they thought it could be a virus, and then they just run the samples through gradients, and then they could isolate. And then they found somewhere they found out there is some virus in it. They do not know at that point what kind of a virus or what it is, how it is going to be. But since they had an access to 200 kV electron microscope, they just put it and imaged it. And within a couple of weeks, um, not even actually within a week, they were able to partially solve the structure to 2.9 angstrom to 2.9 angstrom in full structure and 3.3 angstrom with a capsid, chest capsid. So with this, actually, they were able to partially sequence it just with the proteins. They were able to see the protein side chains. They were able to assign what could be the sequence. So in some sense, they are overtaking the speed of sequencing itself. So Cryo-EM has become more user friendly than ever before. So there have been constant publication development has been happening from single particle analysis. Now people are so as you know, many Chinese and Asian institutes are actually heavily investing on this Cryo-EM. So people are quite interested in shifting to cryo-electron tomography, and currently it is picking up its phase, which I'll be explaining what is cryotomography in the upcoming slides. Mm, and it's picking up a phase, and people are hoping that it will start having its own resolution revolution within somewhere around 2025 or below before that. Actually, it started somewhere around 2020 itself. So you will constantly see this cryotomography, then cryo-electron microscopy in future also future. So what do we see in cryo-electron tomography is, so initially what in electron microscope you see individual proteins or biomolecules. Now people are interested to see how this individual molecule is sitting within the cell, and how the other molecules of this biomolecules of the cells are interacting with every molecules. And you can see the beauty of entire life interaction is happening within the cell at a single snapshot. So this is how, before which we used to see like a GFP, RFP or something, you put it inside the cell and you use a fluorescent microscope and then you see the image. But now with the cryo-electron tomography, you would be able to see all the structures at the same time, like nucleus, ribosome, endoplasmic reticulum, and everything, proteins, majorly of somewhere around megadaltons, you would be able to see and create a 3D image of that, of the entire cell itself. So that is eh, for life science people, it's quite interesting because they can see everything everything all at the same time in one snapshot. So this, if you take cryolotron tomography, this is what the kind of image that you would be getting. The small blue ones are, I think it's ribosomes, and that's I think this is okay. So I'm not pretty good at this reading. Okay, pretty good at understanding this, but the other studies would explain you better. So this is what the cryotomography works. Wait, wait, wait. Okay. Um, okay, I'm sorry, no, I'm not able to show that video. Okay, the cryotomography is like this. So we take the in this case we have used a virus particle, where you can directly put the virus onto the electron microscope, and then you can rotate it, and then every single protein molecules on the virus can be imaged by tilting, and then you can create a tomogram, and then which will eventually gives a cryotomography image of the entire virus itself. So this is the virus. As you can see, you can directly see the membrane proteins on the virus, as well as the entire entire virus at an atomic resolution. Here, the final resolution what we got is like 4.2 angstrom resolution. So imagine yourself as a virus. You can take a picture of you, and then you are actually seeing every single protein on your hand or a face or an eye of sorts. That's the kind of powerful tool cryo-EM can offer, cryotomography. So all these things, uh, in case of virus, it's quite simpler. So when we develop a drug, you can actually see it, how the drug interacts with the protein molecule or the biomolecules. While it is interacting, what kind of impact it creates on the whole virus or a bacteria, it can be studied at a single event, which will very important for most of the biologists. So this is the kind of uh, so this is the tomogram that you would generally get. You make, you take the sample, and minus 70 to plus 70, you just tilt and collect, shoot the electron beam through the same way as electron microscope and tilt it, and then you average all the images, and then you will get a beautiful atomic resolution image of the entire virus, with its all its functional biomolecules intact to the virus itself. So this is one example of virus where you could actually see every single building blocks of the virus itself. So since we are running a little bit low on time, I would just skip that videos. So these are the general images that you would get for viruses. And when you want to go to the tissue samples, all that you have to do is you freeze the sample, and say you want to focus on a specific regions. Let's say you are having a brain tissue where you want to study a signaling pathway. So you put a GPCR or green fluorescent protein or a GFP, RFP or YFP, something, and then you take the tissue and see the place where it is fluorescing, and then you mill it in the sense you just um, etch it so that you remove and make a small thin film of the sample, and then you can put it in electron beam so that you can get the structural analysis. And I will just show you this one place. It doesn't play the video, so I'm sorry about that. So this is how with robot this is a robot which is helps you preparing the vitrified sample, that's freezing the sample, just quickly freeze the sample, and then you put it on a fluid milling where you can actually mill the sample, and then you put it on electron microscope. So you would get something like an image of a eukaryotic, in this case um, eukaryotic nuclear organization. You can see there's membrane proteins that you can see as a pores that you can see, um, and the endoplasmic reticulums, ribosomes, most of the major molecules in a few around like 10 mega about say 50 60 kilodalton all the way up to 10 megadalton, whatever the structure is there. So you are able to see at a single snapshot. So as you can see on the lower right bottom, you can see all the video how the reconstruction of the image has happened. So how this is being done is you take uh, grow the cells on a copper grid, and then you kind of identify a good region of the tissues, just like these holes, like the holes, direct holes, you will just ignore, but those places where the tissue samples are quite intact is just cut it off with with a plasma beam. Plasma beam is like you may take a gas and charge it to a very pass it through a very high electric field, so it gets charged, so the plasma is formed. And then once you uh, focus it on a tissue without spoiling the tissue's integrity, it will just make remove etch the entire samples and leave you with a thin film of lamella. And all these things, if most of, I don't think it is possible currently with manually to do the lamellas preparation. And if you do, you would do something like ultra microtome, which will have few micron size, but this one you can get the film up to a range of nanoscales. um, so what happens is uh, so these are all completely automated. So the software actually helps you in identifying good regions automatically. It will set, and the moment you give the automatic patterns, it will identify good regions, and then fluorescent regions, and then around that region it creates a mask, and then um, cut off the sample and create a thin films which you can use it for imaging. So since we are running out of time, I will just show you some of this. Here is a lamella preparation of chlamydomonas. So the chlamydomonas is quickly frozen, and then it is um, stuck to the copper grid, And the blue, small faint blue that you can see is the mask that is computer generated. So you can pass the plasma and you can create a small thin film of it, and that is loaded onto the cryos. That's 300 kV tool, and then you can get an image of that chlamydomonas. And how does the chlamydomonas looks after reconstruction? It's the whole picture. So as you can zoom in, you can see the nucleus and mitochondria, endoplasmic reticulum with its proteins intact to it, ribosomes, everything you can see with one single snapshot. So these are the um, how you are able to identify the regions. As you can see, you can once you have a GFP or RFP in a system, you can actually do a fluorescent scan, which will help you to identify where the cells are, and you can create a mask and basically you fib mill it, and then you can load it onto the cryos, and then you get a 3D reconstruction of large protein molecules. So uh, to actually
The
so
thermoscientific Oncolos 2 cryofib with IFLM
this
correlator
science,
system
this
combines light
video
and
shows how
electron
it can be done.
microscopy into one instrument. In the system, fluorescence data can be overlaid on SEM images, allowing the localization of fluorescently labeled cells. With integrated fluorescence, you can monitor fibrilling to ensure cellular target regions are within the final lamella. Fluorescence integration gets rid of an extra transfer step, making correlation between light and electron microscopy easier and faster. Search less, discover more. Illuminate your cryotomography workflow with the power of integration.
So basically, it was like this. There is an SEM, that's a scanning electron microscope, which creates a very high resolution. So this ion is the plasma ion fib milling system, which helps you. So that there is a light microscope as well, and then it's a fluorescent microscope, which can, while you can see the fluorescence, you can see it through the SEM and create small lamellas which can be used for high resolution imaging. So these are the basic applications or like developments happening in fluorescent cryo-electron microscope, which is helping to most of the researchers. And some of the researchers who have been still doing crystallography, they are also shifting to micro ED, which is like they take the crystals and put it in the path of electron beam, so you get a diffraction patterns, which is very similar to X-ray diffraction, and solve the structure very similar to that of X-ray diffraction. So you get one tool which is kind of um, satisfying the usage of fluorescent microscope, um, structural biology, cryo-electron microscope, or like replaces tools like X-ray crystallography or NMR and stuff like that, and then it just kind of supports everything in one tool. You get it. So these are the tools. So sorry, I'm just little bit running out of time, so I'll just quickly go through this. So this is the cryos tool, which is I've been saying. So this is the feg electron feg gun. That's a source that we use. Selectors is nothing but a bunch of coils which has been completely motorized, which can select after the once the electron beam pass through the sample, you will get a very diffracted beam of electrons, uncoherent beam of electrons, and before it falls on the detector, we need to select a region or like a specific energy, because if you take all kind of energy, you are going to get a very blur images. The noise is going to be very high when you see the image, but the selector is kinds of filter out um, unwanted or like filter out very specific regions of electrons, which is less than 10 electron volt. So you get a very high signal to noise ratio images. And the Falcon 4i is the direct electron detector, which has which capture all every single electron event. So you get the best resolution of uh, the biological molecule. So most of it can be understood. How Thermo Fisher can help is like we have EM University, where you have complete description, or like how electron microscope work, how it can be used, how it can be studied, and all the way from EM learning to all the way to the demos and the hands-on. We are part of it, and that comes concludes my talk. If there are any question, I'll be happy to answer it.
Thank you, Kumar. That was really an interesting, enlightening talk, actually. So that forms the final for this lecture series. If there are any other questions there, please, this is the right time to ask our guest speaker. Yeah, Dr. Prem has a question, actually.
Yeah, hi Kumar, nice talk. I have basically two questions. One is this correlative microscopy, whether Thermo has the full package, or we have to buy some from like other Leica or.
So currently, the Aculos and Hydra Bio are the recent ones. So once you have that, you don't have to buy any other tools from other vendors. So it has its fluorescent beam integrated to it, so you don't need other clem tools also.
Okay. Second is, so what is coming next, means what we can expect in next 3 years
Okay.
from Thermo.
Okay.
Hardware, software.
So one recent thing that I've been following it up from Thermo side is they're working on laser faceplate. Um, uh, uh, there's some concept called faceplate development is happening. They're investing to increase the contrast of the sample pretty well. So that's what they're focusing on. And there are also some very preliminary studies, or like basic studies have been going on quantum level, how to manipulate the electron beams. So that would be maybe in another 2 to 5 years, 2 to 5 years, I can guess. So that might improve the resolution of perfecting close to 1 angstrom or even beyond that.
Interesting.
So that might be the future, I hope.
Thank you.
Thank you.
Yeah. Can I have one question? Dr. Kumar, as you know, most of them participants here are students. What sort of career opportunities do students have along this field in the industry? And academia, yes, they go for higher studies and all that. But in the industry, are there opportunities for students?
First, I might be little bit negative on that. So please don't take it. But let me say, in India, the engineering part of the electron microscope is not happening at a high level. Like maybe like few material science people. So a lot of industries nowadays, they are using electron microscope, material science wise, but directly life science wise, very few pharmaceuticals have started investing. But by the time the students graduate, I'm very hopeful that most of the pharmaceuticals will be using electron microscope as their primary tool for studying um, uh, pharma products. That's one thing that most people look into in industry. And the second thing is, academia is, of course, majority are actually looking into electron microscope. Other than that, if engineering is engineering is somebody's interested, European countries, especially in Netherlands and Switzerland, they are the one who is actually working on the engineering part of developing a best cryo-EM tools. So Cryo-EM, of course, if somebody has to go through, then Thermo Fisher is one place where they will be on a regular basis posting job opportunities on their website. People can apply through that, and for engineering, they would definitely look for like mechanical engineering, electrical engineering, and stuff like that. But moment it comes to the application, they will look into what are the biology background they are coming from, especially from biotechnology and um, especially from biotechnology. Um,
And
um,
how is the scope for bioinformatics students?
so there's a quite a recent development is happening and in India actually speaking. So there's like with the recent development in AI, a lot of people are trying to use the AI to integrate agriculture, pharmaceuticals, computer engineering, and everything together through life science as well. So they have a pretty good opening in upcoming years. Even now, I've been asked by some of the companies like if there is somebody is there in cryo or is this bioinformatics who's good at coding, who understand the biology, uh, they are happy to invest in them. Um, but only thing is, you cannot just be a good coding person. You need to know the biology. As long as the biology is strong, the coding part comes much easier.
True.
So,
Yeah.
but it has a very good scope.
Thank you.
Thank you, Kumar, once again. Actually, it was really, I'm more than happy to hear you, actually. Yeah. So, with that, we will
Just one
conclude.
thing before
Yeah.
we leave, I'll just say for those people who are interested in career in industries, the best way to go through is not just by applying to the websites. If you just keep on applying to the websites, it goes through a process called HR, and after which the actual people are going to interview you. So the HRs generally wouldn't be interested in sorting the CVs, so they will not be taking up the CVs. So the best way is to contact people who are in the companies through LinkedIn and talking through the making a network. That would be easier for you to actually make a connection. And moment the connection is established, those people are going to intimate or like they're going to ask the HR to look, there's a CV that has been come, why didn't you send? So immediately the HR will forward it. So the first step, that's a HR that's going to be, you have to overcome through networking. Without that, almost 95% of the CVs would be just not seen at all. Although most of you all will be very well qualified. Okay, that's if somebody is interested in industry job, it is pretty good as well.
Yeah, that is the hard truth, actually, unfortunately. Yeah, networking is a key, actually. Yeah. So I hope our student friends, they might have really enjoyed the talk, actually. So if there any one or two feedback is there, then we would be happy to listen to them. Can you please unmute and share your feedback? Okay.
So I'm putting my email on um, for people, students who can contact me regarding any questions on, or anyone who has any questions on this Cryo-EM products or how it functions or to know more about it. Feel free to contact me. Anything all the way from business all the way to the application, I will be happy to answer it.
Kumar, actually, I was thinking maybe we would love to host you in person. Actually, if you find any time, you are most invited to visit campus in Shastra as well as in NIT Warangal. So it will be a pleasure to host you. In fact, actually, and
I'll,
second thing, yeah, is there any kind of industry sponsor programs which you are offering for the institutes like us?
okay. Um, um, Unfortunately, Indian Thermo Scientific is going through some kind of, because of the big boom that has a financial boom has happened for Thermo Fisher during the COVID time. Uh, their their margin of profit has reduced in the current year. So
No
they are
problem.
not creating all this. Initially, there used to be a lot of internships and stuff that used to happen. Nowadays, mostly this is happening at Global Thermo Fisher only. So you might have to look into that. But if something comes up, I can share it with you. But within India, it's very difficult to get an internship at this point.
Okay. Thank you.
Yeah.
Yep. Thank you once again, actually. Yeah. Vignesh, if you want to conclude, actually.
Yeah. Okay. Thanks very much, Dr. Kumar, Dr. Prem, and all the speakers who were there today, sharing their insights, experiences, expertise with all the student
All this.
participants. That's great. Thank you very much.
Yeah.
And so we will be sending a feedback link to all the participants. So please fill in the feedback. That would greatly help us to improve this seminar series in the future. And we also plan to continue a different version of this Cryo-EM workshops or seminar series. So please watch out for us. And I really hope that this sort of gave you all the needed introduction and the depth in which Cryo-EM is making an impact in the research landscape. And I'm sure you will take it forward as researchers or industrial R&D or whatever, in whatever capacity you can. So with that, I thank again for joining us today. Thank you.
Yep. Thank you. Yeah, I think Dr. Prem is also with us, I think, actually. Yep. So he is like from the morning, he's being with us. So it is really nice to see all these busy people taking out their time to share their insights about this exciting field with all the beginners, actually. So I hope like they might have like lightened a couple of bulbs among the student community, and hopefully like some of the students may take up Cryo in the for their future, actually. So thank you once and all. Thank you, Dr. Prem. It's a really pleasure to host you, and thank you, Dr. Kumar, so much for taking out your time to be with us. Thank you.
It's my pleasure. Thank
Thank
you.
you. Thank you,
Thank
everyone.
you. Yep. Thank you. Bye. Okay, I think we can sign out now. Yeah. Yep. Yeah, Biswajit, you can want to say something? I think the seminar series is over. So, yeah, thank you all for participating, taking out your time. And if you have any queries, then you can drop a mail to me, and the feedback link will be shared with you. So in that, you can share the feedback regarding the seminar. If you want to add up anything, this is the right time. You can unmute and share your experience. Otherwise, we will conclude the session.